Welcome to today's webinar. It's an ongoing series by the Institute of Pathogen Genomics, which is part of the Africa CDC. We've been running this webinar since last year, and we run them every 2 weeks or bi-monthly, and we invite speakers of note to speak to us on various topics related to pathogen genomics, from bioinformatics to ethics to communication to genomics. And today we are excited to be joined by Dr. Vinod Skaria. Dr. Vinod Skaria is a clinician and computational biologist. He's the principal scientist at CSIR Institute of Genomics and Integrative Biology in Delhi. He's also a professor at Indraprastha Institute of Information and Technology, also in Delhi. And so at this juncture, I want to warmly invite Dr. Vinod Skaria to take over. Dr. Vinod, welcome.
Thank you very much, Joseph, for the introduction. Over the next 30 minutes or so, I'll give a brief idea and overview of what we learned from the genomic surveillance programs in India. And before I start, let me give a minute to introduce where do I come from and what do we do. So I come from the CSI Institute of Genomics and Integrative Biology. It's a national laboratory under the CSIR system in India, and one of the premier institutes in the area of genomics in India and this part of the world. And typically what we do in our regular life is to work with a number of partners. This could be clinicians, this could be diagnostic labs, this could be industry, this could be educators, and this could be public health personnel, so on and so forth, in trying to evangelize genomics in terms of its clinical and translational applications. And coming very specifically to what I do in my lab in IGIB, we work in the area of genomics and genome informatics, trying to sort of find applications in a variety of areas. And one of the major areas that we work on is in the area of Mendelian genetic diseases, or otherwise called as rare genetic diseases. And trying to extrapolate this in terms of biology, trying to look at the regulation of the genome and also the genetic epidemiology of such diseases, human diseases across the world. And it is only incidental that we used to work on a variety of pathogens very specific to India, like tuberculosis. But with COVID, we ventured out into looking at the COVID genomics and also setting up genomic surveillance programs in this part of the world, not just in India, but also many adjoining countries. Of course, you could find more information about what we do in this website out here. Now, when we talk about genomics and genomic surveillance of pathogens, we need to realize that we need to really go back to a bit of history. And the history of pathogens, or for that matter, even microbiology, is centered around this gentleman, Anthony von Leeuwenhoek, who was a scientist by passion and a merchant and a municipal official by profession. Now, what he invented was this microscope, or a handheld microscope, through which he could observe the microscopic world around him. And this really was the starting point of microbes and microbiology as we know today. Now, it has been 400 years, but still the concept and the key central role of observing the microbes under the microscope seems to have been the cornerstone. Now, of course, in parallel, there was a different kind of revolution happening across the world. And this revolution pretty much accelerated in the last decade or a decade and a half with the sequencing of the human genome through a large multi-institutional, multi-cultural and multi-continent program. And what really happened largely in the last one decade has been the sort of democratization of sequencing technology, that you could now sequence human genomes in very small labs like yours or mine, and in a quite affordable cost and a throughput that is clinically viable. So it is just incidental that we are in the right place, in the right point, that we sort of merge and meet these two sort of landmarks in terms of microscopy improving its capacity, and of course the genomics revolution, which is touching every aspect of human life today. So when we come to this pandemic, in contrast to the other pandemics that was there before, or other epidemics that are there before, the identification, characterization, and observation of the pathogen under a microscope, of course, not the Leeuwenhoek microscope, but advanced microscopes, like, for example, an electron microscope, could take considerable amount of efforts, considerable infrastructure requirement, and of course, more importantly, skilled people to do that. And therefore, this was limited to a very small or handful number of laboratories, even globally across the world. Now enters genomics, and you see that from Wuhan, and the first genome of the SARS-CoV-2 from Wuhan. And if you really go back to the paper which describes this genome, you will realize that it didn't have anything to do with microbiology. What was really done was to take the sample from the bronchoalveolar lavage from the patient, direct nucleic acid extraction, sequencing, and analysis, right? It never went through any traditional processes of culture, isolation, or observation under the microscope. Now, of course, this method is fast, is cost-effective, and more importantly, it is democratic in the sense that anybody who has a sequencer today can pretty much do this kind of an analysis to identify new pathogens out of epidemics. And more importantly, in contrast to the traditional ways, it allows for a direct molecular analysis, that is detection along with a genomic characterization, and all in an affordable cost, in an affordable turnaround, and in type of infrastructure requirements much lesser than what was done previously, or what was available previously. And of course, this is therefore a landmark in terms of applying genomics at scale. It is not just one genome anymore. Today, we have GSAID, which has close to around 7 million genomes of SARS-CoV-2. Now, one thing that we need to keep in mind while looking at genomes of any pathogen for that matter, or for that matter, any organism, the very fact is that these are all pathogens or organisms, and organisms evolve by accumulating genetic mutations. The only sort of distinct differences between many of these pathogens, in this particular case viruses, is that they have a very distinctly different rate of accumulating genetic mutations. They could be very slow, as in case of herpes simplex, or quite fast, as in case of HIV. Now, SARS-CoV-2 has pretty much a constant rate of mutation, and this has been very well documented and discussed quite in the past. And this has quite a number of advantages in its application for molecular fingerprinting. And this sort of signifies how this molecular fingerprinting is done. So every new substitution occurs in the background of an existing substitution, and this rate of substitutions that occur in terms of the counts or the numbers of substitutions is pretty much constant, and also depends upon the spread of the infection. Of course, they may not be constant all across the pandemic, but pretty much constant across a particular point in time when a variant emerges and spreads. Now, as you see from this particular graph, a particular variant is essentially nothing but a combination of genetic mutations. And if you take random 6 genetic mutations, they could occur in multiple different combinations. And the occurrence of the combination would also tell what the immediate ancestors were, and also be able to compute how distant in time was that immediate ancestor. Now, this is very important because in a constant rate of evolution, what you would expect is to have unique proportions or equal proportions of each of these variants. But in real life, we don't see this. We see one or the other types or combinations of mutations which are predominant at a given point in time. And this is typically what we call as emergence. The emergence, of course, could happen because of multiple different reasons, the commonest being human factors like superspreader events, which could transmit the same virus with a fingerprint to multiple individuals, and therefore skew the proportions at the time of genomic surveillance. The other obvious factor is the viral factor. In other words, a virus which can transmit much faster could have a larger than a normal proportion or expected proportion at a given point in time. And this essentially forms the basis of all that we do in genomic or genetic surveillance. Now, when you do genomic surveillance, you will also identify viruses which are close together and seems to have diverged at some point in time from a common ancestor. And therefore, they occur in clusters of very similar genomes, which are similar and have shared genetic mutations. And this is what we call as lineages or clades of the virus. And of course, there are multiple different ways of annotating these clades and clustering these clades. But the commonest of them that's widely used in terms of the visualization is the one that is on next strain, which sort of catalogs the different lineages across the world using a nomenclature system depending upon the time of origin of that particular cluster. But of course, a much more commoner way of naming these clades has been set up by Pango. This is a network of researchers from across the world who put together a nomenclature system which sort of signifies its immediate ancestor and immediate lineage. And it's a very dynamic system that you can keep annotating a particular lineage as if you were like a numbers in an IP chain. And of course, you have now a large number of genetic lineages which are publicly available and could also be contributed by researchers from across the world. And the other important thing for such lineages is that when you, of course, can look at a lineage or annotate a lineage, you can also create a sort of a virtual ancestor to a particular genetic lineage. These are essentially, in virtue, the first virus that sort of was a common ancestor to a particular lineage. Now, this virtual ancestor in many ways may not have been sequenced because of multiple different reasons, but nevertheless, the time of occurrence of that particular ancestor could be computed back in time depending upon the configuration of genetic mutations, based on the fact that combinations of genetic mutations occur at a very constant interval. And this is what we call as the time to the most recent common ancestor, or the TMRC. Now, this is very important because it allows epidemiologists to sort of trace the origin of a particular lineage. And in many ways, if you have viruses or genomes of viruses from a particular geographical location, it could also tell very important factors about when was it introduced and how did it spread in a particular community. And this has been used quite extensively early in the epidemic. One of the examples is the cryptic transmission, which was sort of unrevealed from the genomes out of the United States. Just by looking at the combination of genetic mutations and by sort of computing back in terms of time what was a common ancestor, you could sort of realize that at a point in time, there was a cryptic transmission of an ancestral virus, which of course was not sequenced, and the samples were probably not available, but nevertheless, you could glean very interesting insights into the early introductions and transmission of virus in a particular community. The real problem, nevertheless, was not the lack of tools and techniques to do this, but really the scale at which genomics could be done. Now, until mid-2020, the ways and means to sequence genomes at large scales did not really exist. Typical labs could sequence dozens of, if not a couple of dozen genomes every week. But nevertheless, that was not the kind of scale that was required for large-scale genetic epidemiology. Now, the real high-throughput ways of doing multiplex sequencing of very large number of genomes required for kind of genetic epidemiology applications were essentially put together by a company called Illumina, which manufactures sequences. And this assay called COVID-seq was sort of FDA approved sometime in mid-2020. We were fortunate to have been one of the first labs across the world to have applied this particular assay in a real-life situation by sort of trying to multiplex large number of samples and also put together the analysis infrastructure to turn around the analysis in near real time. Now, the real advantage of such a system was that it was quite flexible and modular in the terms that it could sequence anything starting from around 750 sequences to around 3,000 sequences in approximately around 24 hours. And of course, we put together a computational pipeline which could churn around the information from, or glean the information from these genomes in another 8 to 12 hours. So in something around 48 hours, we would have been able to get information about what is there in these genomes. So with pilot funding from CSR, we sort of put together this assay in a real-life situation and applying this in association or in collaboration with a number of different states in the country, which had little genomic representation in early part of the pandemic. So we sort of collaborated with around 7 states and ended up sequencing around 2,000 genomes in a sort of proof of concept or a pilot surveillance mode, trying to understand, can we find applications for such large-scale genomes in the country to understand SARS-CoV-2 genomes. And to also keep in mind that this is much before even the variants of concern were even put together. The first variant of concern was alpha, which appeared sometime late of 2020. Now, of course, by doing this kind of a surveillance program, we did learn quite a bit. And one of the things that we learned was also that there was a cluster of genomes colored here in violet, as you see here. And this cluster of genomes did not have much of a representation from across the world. At that point of time, the global sequences were dominated by this cluster, which is in blue, which was called the A2A cluster, which has a very specific genetic mutation called the D614G, which increases transmissibility advantage compared to the wild type out of Wuhan. Now, this cluster was very significant because it seems to have been an offshoot of the A3 cluster here in green, and therefore we named this cluster at the A3 I or the I cluster, sort of signifying that this is a cluster very uniquely present in India. And using this genomic surveillance, we could actually trace this cluster to across different states. And one of the ways we could trace the cluster is to use what we call as haplotype networks. And haplotype networks in simplistic senses are ways of clustering genomes which share a common genetic mutation and of course, divergence patterns, which could be mapped depending upon how divergent are each of these clusters from each other, depending upon the mutations shared or different between them. Now, using such kind of an approach, we could look at this particular genetic lineage, what we call as the IA3I, today called as B6, and we could pinpoint this cluster to a single superspreader event in the northern part of India. And as you see here from the graph out here, the lineage in violet had different proportions across different parts of the country. And all of them seems to have emerged from one single superspreader event and with members from that event traveling to different parts of the state and therefore spreading the variant as we call today as B6, earlier as IA3I, to different states or different parts of the country. And of course, over time, this variant or the lineage seems to have disappeared and have been largely overwhelmed by the A2A lineage, which has D614G. And by looking at haplotype maps across for other genomes which are not part of this particular cluster, we could also realize that superspreader events or local outbreaks sort of encompass a significant large majority of the genomes that were sequenced from these states. And we could also identify such large superspreader events in multiple states in southern and northern India. Just a few examples in here, but of very important mention is this red cluster out here, which is out of Kerala. Now, Kerala is a southern state of the country, which has a quite significant expatriate population. Almost a tenth of the state lives abroad. And with the COVID pandemic, a lot of these people were coming back to the state through evacuation flights, which is arranged by the country. Now, many of these individuals had to go through a very sort of rigorous and laborious, I would say, a long-term quarantine measure, sometimes ranging from 21 days to, in some cases, up to 30 days. And as cases were rising in mid-2020, it was widely believed that these travelers had brought in the cases into the state. But what we found from genomic sequencing and the clusters from the genomes, we realized that most of the cases in Kerala seem to have clustered in just 3 clusters for ease. Let's name it K1, K2, and K3. And these clusters were largely, in terms of origin, traced back to the nearby states. So in other words, a large proportion of the community spread of the virus in the state of Kerala in mid-2020 was contributed by travelers who crossed over from the borders of the state rather than international travelers who brought in the international fingerprints for the genomes. Now, this was very important because the entire policy of surveillance, testing, and quarantine was largely focused on international travelers at that point in time, while the genomic data sort of suggested that what we were missing was essentially tracing and surveillance of individuals who were crossing over from the nearby states, which also had multiple such super spreader events. Now, Kerala therefore took this up and sort of created a very systematic program which did surveillance for all the 14 districts and involved the medical institutions, 12 medical colleges in the state, the regional and public health laboratories, 2 university centers and the district surveillance offices, and also was part funded by CSIR and the state government. And this therefore forms the first very systematic program for genomic surveillance in India and started sometime in November 2020. Now, this was really taken forward in terms of building public policy, building evidence in terms of identifying new emerging genetic lineages, implementing policy, for example, like double masking in April, early warnings for lockdowns and augmentation of capacity, anticipating a wave of Delta, and of course, much recently, surveillance of the Delta sublineages in terms of the clinical profiles, and also surveillance of Omicron as we see Omicron has spread across the world. And of the genomes that are done in the country, close to around a tenth of the genomes have come from this single program from the state of Kerala. Of course, there are other applications than building public policy. And one of the most critical applications for genome sequencing is to sort of evaluate and understand what are the diagnostic primers that are likely to fail given a particular genetic variant. And the principle of this is very simple. Most of the diagnosis, or the gold standard for diagnosis, is based on RT-PCR. RT-PCR requires a particular oligonucleotide to bind and recognize the cognate sequence in the viral genome. And therefore, a specific genetic mutation in the viral genome could impact the efficiency at which this binding and amplification could occur. So, of course, you could use computational methods to understand what is the effect of each of these genetic mutations, because complementarity is defined by thermodynamic rules. You could compute the free energy of binding and, of course, look at genetic mutations, which could significantly alter the free energy of binding of a particular primer or a probe to the viral genome. Now, early we did this analysis trying to understand what are these primers and probes. We could compile them from public and global literature, put together a set of genetic mutations from genomic information that is widely available, and ask this very simple questions. Are there mutations which could impact the efficiency of these primers and probes? And if they do, are they frequent in a particular population? Now, of course, what you see here in the graph is essentially on the x-axis, the genomic location of these mutations, and the y-axis, the frequency of the genetic mutations in global populations. And you would see that there are some genetic mutations which could adversely impact the efficiency of some primers and probes in some kits. And of course, this is very valuable information because then you could inform the governments who are using those kits quite widely. The other application of genomics today is in understanding and differentiating close clinical differentials, like, for example, persistent infections, reinfections, or reactivations, and much more recently, breakthrough infections. And this largely is because of viral mutations, which could escape pre-existing immunity, either from pre-existing viral infections or from the vaccines. I'll give you a few examples for this. The first example, of course, is of these 2 individuals, healthcare workers who were put on surveillance in one of collaborating hospitals. And these 2 individuals tested positive twice in 2020. And both episodes were asymptomatic. They were managed conservatively, but nevertheless, we could have access to this sort of RNA from this test. And genome sequencing from the two episodes, E1 and E2, for these two individuals, I1 and I2, sort of suggests that these two genomes are quite distinctly different in the mutational fingerprints. In other words, this sort of can molecularly establish that these individuals indeed have a reinfection by a different virus, which is a different molecular fingerprint. And this probably was one of the first reports for asymptomatic reinfections in global literature. Now, a close clinical differential to this is individuals who could have 2 rounds of RT-PCR positivity with an intervening RT-PCR negative status. This could also be a virus which is hidden deep in the body, and that is what is called as reactivation of the virus. Now, reactivation of the virus is not very well documented. There are only a handful of publications. But how do you differentiate a reactivation? And that's because the E1 and E2, or the episodes E1 and E2, would exactly have the same mutational fingerprint. Though they are spaced apart from a period of RT-PCR negativity, they could pretty much be the exact same genomic or mutational fingerprint that's present in 2 episodes. Therefore, you could differentiate reactivation and reinfection just by sequencing the samples which are available for these individuals. And of course, a much more recent entity is what we call as breakthrough infections. And breakthrough infections occur in individuals who had or a full dose of vaccine or immunized by 2 complete doses of the vaccine. And this was also from a cohort of healthcare workers who were on surveillance and who also had access to early doses of the vaccine. And what we realized is that many of these individuals turned RT-PCR positive on regular surveillance at a later point in time. And this also therefore could contribute in terms of the genomic understanding about the variants of concern or the early variants of concern and their capability to produce breakthrough infections. Now, with of course, with Omicron, breakthrough infections are commonplace, but this was at a point in time where we thought that the 2 doses of vaccines could pretty much protect us against infections also. Now, of course, these are the applications, and what I'll spend the next few minutes is sort of delving deep into where do we go from here. And to understand where do we go from here, you also need to understand how did the virus reach what we have seen today. And what we have seen recently until recently was Delta, and much more recently was Omicron. And the virus seems to have evolved in very consecutive steps and sort of traversing this landscape of transmissibility as well as immune escape. And at each of this evolutionary steps, which have created either a variant of concern or a variant of interest, it has sort of traversed this space, trying to improve in either immune escape or transmissibility, or in some cases, or rather most cases, both of these capabilities. And therefore, you need to have resources to be able to understand how is the virus doing this, or what are the mutations with functional consequences which can enable the virus to do that. Unfortunately, we were one of the first groups to have been compiling such information in a very systematic format, and there are 2 databases which are probably the most comprehensive ones today available on immune escape as well as functional genetic variants in SARS-CoV-2. And using this information, you could sort of understand, looking at the genomic sequence and the mutations from the genome, what is the virus trying to do. And of course, that's when we realize that the virus indeed has an Achilles heel, and the Achilles heel is indeed the spike protein. And that's pretty much because the spike protein is the protein that the virus requires to latch on to the human receptors, or the ACE2 receptors, for infection, and is also the exposed protein which needs to be recognized by the antibodies, or rather, which is the protein which is recognized by the antibodies. So in other words, the virus has only a very limited number of genetic mutations which are possible, which can evade antibodies, but at the same time protect its capability to attach to the ACE2 receptor. Now, these 2 are not mutually exclusive, because if it loses its ability to latch onto the human receptors, then it cannot pretty much infect the human, the host. But for example, if it cannot escape the immunity, then pretty much the antibodies will coat the surface of the spike protein, and therefore it cannot latch onto the receptors. So in other words, there are only a finite set of mutations, and this finite set of mutations could occur in multiple different combinations, and that's what we have been seeing for the newer variants which are emerging, whether it's Omicron or its sublineages. The virus is pretty much trying to accumulate one or more of these mutations in combinations. And that came in quite handy with the Omicron, because the moment we had seen the Omicron sequences, the first 7 genomes in public domain, we could pretty much understand what is this virus trying to do in terms of immune evasive capability as well as transmissibility. And it's not a surprise that Omicron became a variant of concern in probably around 72 hours from the point in time that the lineage was described in public literature. And that is pretty much because of the fact that the confidence that we can associate with these genetic mutations and their ability to evade the pre-existing immunity is quite large. And therefore, in the future, as we see more and more Omicron sublineages, we sort of add to this confidence in our ability to understand what is the virus even trying to do. The other important point of that we need to keep in mind is to understand when and where should we do genomic surveillance. And in practice, what happens everywhere across the world, India is not an exception, is to sequence more when you have more cases. And in fact, what I'm trying to show here is that if you sequence more when there are more cases, it's really not going to add much to the diversity or the understanding, because whatever is a successful variant is what is going to cause a wave. And pretty much what you get from sequencing at a wave is essentially 1 or 2 types of the same variant. But the diversity is largely captured during the trough phase of a pandemic or an epidemic. And as you see here, this is the wave, the second wave in India. The outbreak started with small outbreaks in Yavatmal and Amaravati. And as you see here, if you had quite significant coverage of sequencing during the trough phase of the wave, you could have captured many of these variants much early, because as you see here from the computation, many of these variants emerged during the previous wave, or rather the first wave of COVID in India. And it's understandable because variants emerge when there are large number of infections, and that's when the mutations will happen. They lie low until they have a conducive environment. So in other words, when you do sequencing for surveillance, it's very important to consider sequencing quite enormous number of samples or larger proportions of samples during the trough phase of a wave, rather than focusing all your efforts into sequencing more and more when the cases really go up. And to identify many such events or outbreaks, you need to have fast sequencing turnaround. And we have put together one of these protocols which can work on benchtop equipments, which can sequence today anything from 48 to 96 samples in less than 24 hours. It also comes with the informatics solution to be able to provide the analysis and interpretation of these genomes all within 24 hours. And this has been now widely used across the world, also in association with the WHO Southeast Asian Office for surveillance for many nearby countries, including Nepal and Maldives. The other important question that we need to keep in mind is that we do have a lack of representation of genomic sequences from across the world. And that is also because we rely on a centralized way and a policy for genomic data deposition. What we probably need is a much more democratic and much more federated system, so that many of these data sets which are otherwise sequenced, could now be shared across the globe for interpretation and analysis. And one of these frameworks that has been widely used for genomic data, human genomic data, is the Beacon framework, put together from the Genomic Alliance for Global Health. And this is the dashboard for SARS-CoV-2 genomics or genomic surveillance in India. And we are probably one of the first dashboards to enable the Beacon, which enables programmatic access to the entire data set, including the metadata for analysis and interpretation. Nobody needs to really download the data and reanalyze it. You can programmatically access it using standard protocols, which is specified by the Beacon APIs. And of course, we are now enabled beacons for many of our resources, including our databases, including ESC as well as Favicov, so that other resources and genomic centers can access up-to-date information in real life without having to re-download these resources over and over again. So whenever the database is updated, the same information is also transmitted through the beacon through the analysis software that uses data. I think I'll stop there and take questions, but before that, a bit into how we see the future of genomic surveillance. We are probably in the fortunate period in time because there are few concepts which are dominating the discussions on genomic surveillance. One and most important thing is that we have today digital information which can be transmitted at the press of a button. We have increasingly used genomic applications globally to understand such outbreaks. And even more importantly, the globe has realized the need for openness and collaboration, because a pandemic of this scale is not a problem of a single country. It is a global problem which can affect everybody. And of course, this requires data to be computed and integrated at multiple levels. And fortunately, this data is all digital and available quite readily. What we really require are frameworks for digital integration and frameworks for collaboration, as well as sharing of this genomic information. What we've been trying in the lab very recently is the flu map, where we've been tracking multiple diverse sources of information, like from migratory flybase, weather information, news, social media, post genomic fingerprints, to try to predict how is avian influenza H5N1 spreading across the globe. I think I'll stop there and take questions, but before, let me acknowledge a lot of people who make this possible. Fantastic students in my lab and in the lab of Dr. Sridhar Joseph, who has been a quite significant collaborator, and of course, clinicians from across the country and now across the globe, and funding from CSIR through the initial grants and later grants from the state programs, the DBT from the Government of India, as well as the WHO. Thank you very much.
Thank you very much, Dr. Vinod Sukaria, for your very detailed presentation. Of course, starting off with the introduction of pathogen genomics and the context in India, but also your deep dive into the SARS-CoV-2 scenario in India. So participants, for the next 20 or so minutes, we are going to take questions. You can post your questions on the Q&A box at the bottom of your screen. We'll be reading them out, and Dr. Vinod will try and answer as many as possible. My name is Joseph Wangendo from the Institute of Pathogen Genomics, Africa CDC, based in Addis Ababa. So I'll start off by the first question on the Q&A box. Dr. Sukaria is, thanks for the nice presentation. Can genomic sequencing be used to predict variants of concern rather than detect them when they are already in circulation?
Yeah, thank you. So the quick answer to the question is we are not able to predict what a variant of concern would come up, but looking at a genome of a potential cluster of what you call SARS-CoV-2 from an outbreak, we would be able to make a quite useful guess on what this virus is likely to do. And the best example of this is Omicron, because the Omicron lineage was assigned as a lineage with just 7 genomes, right? Typically, a lineage is assigned with hundreds of genomes, but Omicron was assigned with just 7 genomes. And this came out of Southern Africa, from South Africa and Botswana, as well as travelers to Hong Kong. The point is, we didn't have any additional information other than the genome sequences, but nevertheless, with a priori information about what these mutations could do, you could put a fairly good guess that this indeed is something that is going to be a variant of concern. Now, that is the power of genomic surveillance and integration of data and resources to enable interpretation of genomes. Now, we may not have been able to predict Omicron. Nobody indeed would be able to. But once you have the sequence of a particular genome or a variant, we pretty much would know what this virus is likely to do. So that, in many ways, is what we do. We pretty much cannot predict what will happen in the future. We can get an informed guess of what has happened.
The next question is from Nandini. Why is there a wide variation in the sequencing data reported in the press and the actual GSAID uploads from India? Over.
Yeah, so the quick answer to that is GISAID is a public database, but in many ways it is not a democratic database. So there are many countries, there are many programs which don't default upload it on GISAID. There have been multiple issues with that, of course, issues with the reuse of data, etc., etc. I'll not delve deeper into that. Therefore, there are programs which do not confer to the restrictions and the rules of GISAID and therefore do not upload it on GISAID. And for that matter, GISAID is not a democratic database for that matter. And that's why I sort of specified why countries would require federated resources without losing their ability to share the data, to interpret the data, and use the data in ways they deem fit, rather than be restricted to a centralized way of depositing data.
Next question is from Dr. Bardini. How far is it advisable to take an RT-PCR test for COVID within 24 hours?
I think I would apologize that I would not be the right person to take this answer. But of course, we could discuss on this and the evidence probably offline without losing the focus on genomics and surveillance.
Next question is from Vishnele. Is there an indication that Omicron is establishing an animal reservoir?
Well, there is nothing like a conclusive proof which sort of suggests that. And it's unlikely to be confirmatory in any aspect, because though there are a number of animal reservoirs now, or an animal intermediate host now identified for different SARS-CoV-2 lineages, it is pretty much difficult to sort of understand what are the mutational fingerprints that each of this host enable the virus to evolve towards. There has been a collection of such mutations, but these are probabilistic models. These can never be absolute models to establish that this indeed was the animal reservoir. So this still is an active hypothesis at this moment.
Next question is from Richard. How much capacity is in place for genomic sequencing in India? What advice do you give to resource-limited settings on the use of genomic sequencing and surveillance?
Okay, so the Indian capacity for genomic surveillance is quite established and quite large, largely also because of the scale-ups in sequencing and analysis capability that happened in the last 1 year or so, thanks to the national program called Saskov. And that's also because India had a very large capacity of manpower and resources in terms of skill sets, which are widely available across both the national institutions as well as the state laboratories. Now, many countries may not have this kind of a resource in resource limiting settings. It is, of course, important to prioritize what we need to do. And therefore, I did put that particular slide to sort of say that if you had very limited capacity to sequence, right, you would sequence more at the trough of the wave, and you could sequence less during the wave. Sequencing during the wave is going to add very little to our understanding about the virus, because you're pretty much going to see an established lineage of the virus over and over again. That's pretty much not of much value, except for the fact that early during the wave, it's very important to understand what is growing. But much more important is to have probably a constant number spaced across whether there is a wave or whether there's no wave. So you can plan the resources. You could sort of stick to an action plan without getting perturbed by a wave and sequencing more during the wave. So I think when resources are limited, the best way is to spread it evenly across the time scale so that you can plan and implement in a very systematic fashion.
Next question is, any insight into the BA 1.1 variant in India?
Okay, so Omicron largely has 3 clusters called BA1, BA2, and BA3, and BA1 has a sub-lineage called BA 1.1. Of the lineages which are widely prevalent today in the country, BA2 is the most prevalent lineage. BA1 has been on its way down. BA1.1 is extremely rare in the genomic surveillance data.
Next question from Rama from India. Could you please let me know why there was lack of genomic surveillance before the second wave?
Yeah, so the quick answer to that is genomic surveillance capacity requires considerable amount of time in terms of coordination and picking up in terms of capacity. The surveillance program in India started only December 2020, so we had only 6 months before we could really learn the tricks of doing genomic surveillance. So it is really unfortunate that during the second wave, we didn't have much of capacity established and running to be able to sequence very large numbers.
Next question from Sumaya. Does the geography of the world have any role in the different mutations of the virus, like the Delta arose in India? Over.
I don't think there is anything to do with the human population or the geographical diversity of the population. I think the most important thing that we need to consider about emerging lineages, I'll not say variants of concern, but emerging lineages are pretty much directly proportional to the number of infections in the particular region. That's also because viruses accumulate mutations during infections, right? The errors happen during the phase of replication. So if you can minimize infections, of course, the lineages or diversity of the virus in that particular geographical region would be minimal. Of course, you could see that in places like Australia, or you could see that in places like Japan. But of course, there are large countries like India or many of the African countries which have very significant transmission that is continuing unabated. And therefore, the probability of finding new genetic lineages are also very large. Now, the probability of finding a VOC is also directly proportional to the number of lineages that emerge, because the virus is essentially trying to sort of sift the landscape of evolution. And if you have many more combinations that are possible, the likelihood that one of them would be successful is pretty much there. So therefore, what's really important is to actually stick to our basics to ensure that the transmission is kept in check. And of course, apart from vaccines, there are non-pharmacological interventions like good mask, ventilation, which could decrease the number of infections. If you decrease the number of infections, the possibility of lineages to emerge is also much lesser.
Question from Govindan. Some mutations are shared between different variants. So is there any possible recombination events in the host?
No, so this is indeed a very good question. Recombination events are pretty much rare in SARS-CoV-2, unlike, for example, the avian flu viruses, where recombination is the mainstay. Now, there are mutations which are shared between lineages. That is because these mutations have some functional relevance and advantage as much as the virus could go. So I would say these are examples of what we call as convergent evolution. In other words, different lineages trying to optimize to the same kind of mutations because those mutations provide a sort of functional advantage to the virus.
Next question from Akondi. Could you identify an invariant part of the virus that could possibly be targeted for vaccination, like the stem region of HA in influenza virus?
Yeah, so it's a very relevant question in terms of looking at invariant regions, but invariant regions are pretty much in the non-spike genes of SARS-CoV-2. The spike protein pretty much doesn't have a really invariant region. Now, that is not of really big concern because this invariant region, apart from vaccines, could be of importance, for example, for building next generation diagnostics, because as I sort of suggested, there are genetic mutations in many of the variants which could evade one or the other primer in a particular PCR reaction. The best example is a spike gene primer in one of the commonly used PCR reactions, which causes spike gene target failure, which is used as a proxy for the BA1 lineage of Omicron. Now, this information can be used to build next generation of PCR test. And indeed, there are many organizations which are already building this based on invariant regions in the genome. Unfortunately, we don't have an invariant region for spike protein, and spike protein is really important because that's what is the exposed protein which could elicit an antibody response. Many of the other proteins are pretty much deep in the virus envelope and therefore may not elicit an antibody response.
There's a question from James Otieno. Everyone agrees that genomic surveillance is a great early warning system for pathogen outbreaks, but this often needs cooperation between labs, data sharing, overcoming bureaucracy, etc. For a country as large as India, how is it all coordinated for a collective goal?
Yeah, so that's a very, very pertinent point that has been raised. So, I mean, of course, in my belief, the real way forward is to have people the freedom to use the data in the way they deem fit. Now, each country would have its own priorities, its own restrictions, its own ways of sharing data. But having said that, it's also important for not just COVID, but a lot of other diseases which cross across borders, like, for example, avian flu. It's very important that countries do collaborate, because it is not really a problem of one country anymore. These are global problems which are going to affect everybody, not just humans, but also animals, spanning multiple different countries. So I think the COVID pandemic has sort of brought people together in terms of the need to collaborate. And that's essentially why I suggest that you always need to have a federated system where countries are free to decide what are the principles of sharing data, but nevertheless conforming to a common goal of what's the minimum set of data that needs to be shared. Of course, there are other priorities. For example, in research environment, these are about publications, these are about credits, but nevertheless, as long as we have a system which is commonly agreed upon, something like a common set of metadata that needs to be shared, I think reaching consensus about this is much easier. In contrast with a central database deciding that if I don't have this piece of information, I'm not going to put the data up there. Every piece of data is important and useful. You cannot create a global system of policing countries on what they should put and what they should not put. I think the country should have the autonomy to decide what they want to share, but nevertheless have a common set of information that is mandatory to be shared. The problem with a common database is that everything becomes mandatory, and therefore people end up not depositing data out there. Now, for a country like India, of course, it's a daunting task because we have multiple states. In India, the state controls the healthcare policies and the healthcare decisions, and in many ways, the healthcare investments. There's a disparity in the capability, infrastructure, and the investments in healthcare between different states. And therefore, there is always conflict. And of course, some states having a lot of resources and some states having less resources to arrive at a common goal. And I think that way the national program like INSOCOG could sort of fill in the void in many ways, because these programs are funded centrally, where the state doesn't have to sort of pay much into running the program. But nevertheless, these programs also have the flip side that there could be states like, for example, Kerala or Maharashtra, which would like to do more because of the advantages that they have, and therefore it might be a conflict with a centralized way of doing it. So in India, both these systems work. So there are programs which are a part of INSOCOG, which provides the minimum baseline data, and there are also programs which are driven by the state, which is add-on to what is already there. So at this moment, there is Maharashtra, which has an add-on program to the national program, which provides a much more comprehensive view of what is happening in the state, so that the state can act upon their policies or create their policies, their ways of managing things, their ways of alerting the public health system, without really having to rely upon much lesser data that is mandated from the national program. So I think both of them are important. And in a federal environment, whether it is nationally or globally, people's advices and people's views have to also be should be taken into consideration.
We have a question here. If possible, could you mention the approximate minimum cost of sequencing per sample?
Yeah, so the cost of sequencing per sample is going to be very different in different parts of the world. This is largely because of both national as well as international policies. So it's very difficult to actually put a price onto this cost of sequencing, because the cost of sequencing is not just the consumables, but also a lot of ancillary factors, like, for example, the basal testing infrastructure, availability of RNA, the transport cost, people and the manpower and salaries involved, etc., etc. And these are going to be very different and distinctly different between different countries. So it's really impossible to really put one cost to global sequencing, but I would say you could put a fairly good range to the sequencing cost in a particular region or a particular country.
We have a few more minutes for a few more questions, but for those of you inquiring, this webinar will be recorded and we will be sharing a link so that you can view it at a later point. There's a question from Christopher here. Are there best practices observed during SARS-CoV-2 genome sequencing that could be scaled up to other infectious diseases, especially in poor resource settings?
So this is indeed a very important point. There is considerable amount of infrastructure and resources that have been generated thanks to the COVID pandemic, because a large number of countries who would otherwise have not gone into the sequencing way have actually gained both the infrastructure and resources to start contributing in genomic surveillance. And at this moment, the genomic surveillance is largely around COVID, but it's also very important for each of these centers to actually go back and sort of chart a plan based on the local need on how these resources are going to be used after COVID. Now, of course, this could be in multiple different ways, which would have clinical implications. There are regions in the world which has high incidence of, for example, HIV, where sequencing is an important component of looking at resistance to therapies. There are regions in the world which sees large number of animal or zoonotic diseases, where surveillance of such zoonotic diseases are important. The regions in the country where, for example, antibiotic resistance or antimicrobial resistance are important, and many of these sequencing resources and surveillance resources could be used for that. Now, as long as we have created a global system in terms of sharing data which is relevant and useful for global good, I think this is something that we, as not just countries, but as a community of individuals in this globe, should think on how to put these resources to future good use, not just for COVID, but also beyond COVID.
There's a question from Esther Nakazi on the chat box. Using genome sequencing, are you able to know if some vaccines are less effective against some variants?
Unfortunately, no, because the vaccine efficacy is largely dependent upon the two arms that it elicits, one is the humoral response, which is modulated by the antibodies, and the other one is a cellular response, which is modulated by the T cells. Now, while the genome sequences can provide quite good insights into what is likely an antibody escape, unfortunately, we are not in at this point in time to be able to predict what it is going to do in terms of the T cell escape. At least for COVID, we do not have a T cell escape mutation or a set of mutations which can completely escape the T cell immunity. And that's also because T cell immunity is a bit complicated, because it also has the human component, which is modulated by the MHC types, and which is diverse in global populations. So it's also very unlikely that there is going to be one variant which could also escape all the T cell immunity. So at the point in time, the genomic information is largely biased towards looking at antibody escape mutations, and we can fairly do that. We cannot really differentiate between vaccines, but we can fairly give an assumption or a prediction about whether it could evade antibodies which could have produced either by vaccination or by previous infections.
I will ask the last question here from Vidya. Is there any possibility for influenza to emerge as there is cases of co-infection of influenza and SARS-CoV-2 reporting? Any chance of recombination?
The chances of recombination are very rare because on one end, such co-infections are indeed quite rare. But co-infections indeed would occur because there are pathogens, different pathogens prevalent at different points in time and different geographies. So co-infections of pathogens are something that is going to happen, and we will hear more and more about these co-infections, because it's also depend upon what time of the year did a particular wave occur, and what was a co-existing pathogen at that point in time during the COVID wave. The chance of recombinations and the chance of recombinant variants to transmit forward are extremely rare. Even if you take influenza for that matter, even for the case of influenza alone, chances of recombinations are not really great. Recombinations are indeed extremely rare events.
At this point, I'm going to thank you, Dr. Vinod Skaria. Thank you for your excellent presentation, and also the fact that you were able to answer most of the questions, actually all the questions. We are really appreciative of your accepting to be a speaker in this webinar series. Like I said earlier, this webinar series are conducted by the Institute of Pathogen Genomics at the Africa CDC in Addis. I've been your moderator. We will have the next webinar on the 16th of February. We will be having Professor Iruka from the University of Ibadan in Nigeria talking to us about the context of antimicrobial resistance in pathogen genomics. So we will share a recording of this particular webinar. We will share a link and you'll be able to access the recording. So once again, thank you very much for taking your time, Dr. Vinod. Thank you all participants for again making time. This will be an ongoing series, like I said, every 2 weeks. So be on the lookout for the next one. And thank you very much and have a good rest of the day. Bye.
Thank you very much.